a supplemental lecture on these two chapters, 17 and 24. And we kind of go into details of these lytic and lysogenic cycles. But if you haven't seen it, yeah, I like to describe it as two different type of horror movie villains. So the lytic cycle is kind of like the hack and slash victim that just kind of decimates, like Jason Voorhees, who kind of just chops his victims up. And it's kind of brutal. And that's because in the lytic cycle, a virus basically goes in, it'll replicate inside of the cell until the cell just bursts full of virus. And then those viruses are kind of going into other cells. So it's kind of this really destructive kind of killer, the lytic cycle virus. The lysogenic cycle is that more kind of mental thriller type of horror victim, the one who kind of seeps into the cell, doesn't actually kill the host cell at first, it actually just integrates with the host cell's DNA. And so now your DNA just has virus DNA with it. So you're expressing everything the virus needs, and then when you replicate, when your cells replicate, the virus DNA replicates with it. So it's a lot less brutal than the lytic cycle, but eventually, your cells will make the virus, and the virus will spread just in a kind of more hidden manner where it's hiding inside of your own DNA. But regardless, viruses rely on their hosts to replicate. And note that when I've talked about the cycles and what I just mentioned, this term replicate, I'm using the term replicate specifically. I'm not using the term life cycle or reproductive cycle. And that's because viruses are in this sort of gray area of life. So when we think of a living organism, what kind of traits come to mind? So just take a moment and think, what makes a living organism versus a non-living organism? What kind of requirements are there in some way? Can anyone think of anything?
Yeah. Not breathe, because there's anaerobes.
It can move under its own power.
Okay, moving under its own power in some way. Okay, yeah, most organisms can do that. Yeah, most living organisms can, have some form of independence in some way. What do all living organisms have? What makes us?
DNA.
Yeah, DNA, right? So we have some genetic code that makes up all the proteins and the genes that encode for every cell that eventually makes us. We're encoded by our DNA. Anything else come to mind?
Reproduction.
Yeah, reproduction. Yeah, a living organism reproduces in some way. I like that you said reproduction and not, necessarily sex, because some individuals reproduce asexually, but they're still living organisms. Lots of bacteria. We'll see even cases of animals and plants that reproduce asexually.
Respiration.
What's that?
Respiration.
Respiration. Okay, yeah. So that's a more technical one.
Because there's anaerobic and aerobic.
Yeah, there are anaerobic and aerobic, but it, more generally, I would say kind of some form of energy consumption, right? Yeah. So respiration, or however they get their energy, right? Yeah. So they use up energy and kind of turn it into something. So viruses, the reason we say they're kind of in this gray area is, first of all, not all viruses even have DNA. They're not even made up of DNA. They're actually, a subset are made up of RNA. Can anyone think of an RNA virus that has maybe been pretty popular over the past couple of years?
You're going to say COVID?
Is it coronavirus?
Yeah, the coronavirus is one RNA virus. So it doesn't even have a genome like we think of it. When we think of our genome, it's all our genes encoded into DNA. Lots of viruses don't even have DNA.
So it's
Some
just
of
one
them
strand?
do, and we call those DNA viruses. But other viruses like coronavirus are just strands of RNA kind of encapsulated in something. And the other aspect, this idea of reproduction and independence, is something that viruses pretty much lack. So viruses rely entirely on a host to reproduce. Now there are some organisms that need a host in order to reproduce also, but the difference is they utilize the host to then reproduce themselves. So they kind of treat the host like a home, and then they're reproducing within that home. With the virus actually doesn't need the host just for the home, but they need the host to make the next offspring. It has to do with the way viruses work in general. And we'll use an example talking about enveloped viruses. So enveloped viruses are just a different subset of virus. So it doesn't have to do necessarily with whether it's RNA or DNA. An enveloped virus can be a DNA virus or an RNA virus. So if we think of a virus, I said it's some genetic material inside of something. And that's specifically, my next Sharpie, but it's kind of the two components that comprise a virus is its genetic material, whether that's RNA or DNA, and the thing that it's in, which we call the capsid. So there's some genetic material, it can be DNA or RNA, it doesn't really matter, and then it's housed in something, the capsid. And an enveloped virus is just something else is around that. Specifically, it's the host cell's membrane. And when we walk through the replicative cycle of a virus, it might make sense why some of these viruses have an envelope. So not all viruses have an envelope, and an enveloped virus doesn't tell you if it's a DNA virus or an RNA virus. So you can have basically, in this case, 4 different types of virus. You could have a DNA virus that doesn't have an envelope, one that does have an envelope, and then the same for RNA. You could have an RNA virus that does or does not have an envelope. But the envelope surrounds the capsid. And it is kind of cool to think of what this envelope does. You can think of it almost as a sort of mask. So our body, we have an immune system that essentially, one of its primary functions is to fight foreign cells, essentially. Cells that aren't ours, recognize what is or is not ours, and if it isn't ours, try to eliminate it. And so what many viruses have kind of developed is this ability to wear the host cell and travel this way undetected. So basically, it'll produce the same exact proteins that a host cell has. So if we have a bigger host cell here, all the proteins that line a host cell's membrane is kind of the code it uses to talk to other members of its own. So basically, if it recognizes that it has this sort of protein, it recognizes it as, oh, one of mine, one of my own. And so when a virus that's enveloped kind of makes contact with a host cell that it wants to enter, the host cell will kind of detect that it's got the same receptors, and then it'll let it in. So this is just the same way cells transport nutrients from one cell to another. Nutrients will kind of leave in a piece of the cell membrane that hits another cell, and that's how you get nutrients from cell A into cell B. The virus just takes advantage of this transport system, and we call that the envelope. It allows it to travel to the next cell. The cell sees that it's got the right keys, the right proteins on its membrane, recognizes it as, oh, this is just one of my cells, it lets it in, and the virus comes in with it. And so specifically what will happen at this point is the only part of the virus that'll go in is that genetic information, the DNA or the RNA. The capsid just kind of helps it finish the entering process. So what happens is the envelope tells the host cell, let me in. The capsid kind of helps break into the cell, and the genetic material will be the only thing that comes inside. So it's just a strand of DNA or RNA at this point. You know, we talked about DNA or RNA being that sort of blueprint, right? It's kind of a recipe. It doesn't actually do anything, right? What the proteins are what do things, right? So the things that destroy a cell or have negative impacts on us is not DNA or RNA, but the protein it makes. So if the only thing coming in is this genetic information, you might wonder, okay, how is this bad, right? It's just DNA or RNA. Well, what happens is anything inside of a cell is assumed to have passed this filter, right? It's already gone through that checkpoint. So anything in here, the cell assumes is its own. And so what happens is proteins exist throughout the inside of this cell, and they're designed to, whenever they see RNA, they kind of, a protein will, you can think of it as Pac-Man. It'll kind of eat up this genetic material and poop out a protein. So it'll convert that RNA or the DNA eventually into a protein that actually does the harmful or beneficial stuff. And so once the virus RNA is inside of the cell, the host actually does all the virus building. So using our own proteins, our own ribosomes will convert the RNA or DNA into the virus proteins. It'll build all the building blocks the virus needs to make a new virus, including replicating the RNA itself. Now when this happens, once you have a fully built virus, it'll slowly kind of float to the exterior of the cell. And what happens is, as it leaves, if we kind of move to this phase, as it leaves, it'll kind of just push its way out of the cell. And as it pushes its way out of the cell, it's carrying that host cell's membrane with it. And the virus is here. And then eventually it pinches off of the cell, and you have this again. So in this whole process, there's no way the RNA in a virus or the DNA in a virus can't replicate itself. It requires the host cell to replicate for it. Yeah, have it.
How did it originally get the scan of the other cell? If it's a cycle and it needs the envelope to enter the new cell, how did it originally get that envelope?
Well, it likely evolved from an unenveloped virus, a non-enveloped virus. So remember, that viruses can be DNA or RNA, and they can be enveloped or non-enveloped.
So it
virus.
evolved into an enveloped virus.
Yeah, so, yeah, it's likely that
By another method
it
of
evolved
entry.
from a non-enveloped virus.
By another method of entry.
What's that?
By another method of entry.
Yeah, so there are other viruses. So this is just the replicative cycle of an enveloped RNA virus. So there are other replicative cycles described in your textbook. I kind of just go through this one as an example, but some, let me see if we have the typical image of a virus here. I'm trying to see on my laptop before I show. I don't have them pulled up in my slides right now, but there are different ways, different forms of virus. One of them is called the bacterial phage. So whenever you see a virus kind of in, maybe the first image you think of is not necessarily just this kind of 20-sided dice, but rather that on top of a long tube right here with almost spider legs. Have you seen this image in some way?
T4?
What's that?
T4?
Yeah, so that's one example of what we call a bacteriophage. Really just, we can think of it as a phage virus. A bacteriophage is just a phage virus that inhabits bacteria. You can have other types of phage viruses. But this is one way that virus is used to get into cells. So this again can be either RNA or DNA. And what happens is, kind of like a spider, it's going to dock onto the cell. And then this you can think of almost as a syringe. It even has basically a sharp structure that punctures the host cell. And then all that comes out. This never enters the cell. Only thing that ever goes in is the RNA or DNA.
So it's like an EpiPen?
Yeah, it's like the EpiPen way to get into a cell.
Or a mosquito.
Yeah, a good analogy there. Yeah, so basically it just kind of injects itself in. So this is one way this is just one example of a non-enveloped virus. They have other ways to go in, but you could imagine how perhaps this might eventually evolve into this. You could see the benefit of this strategy evolving from one of these. So the problem with this is we don't make anything that looks like this, right? So this might be relatively easy to detect as a foreign pathogen. So its defenses are probably in trying to stay alive even though the host cell knows this is a foreign agent. It's very hardy and hard to kill. So it can just endure a lot of what we throw at it. But with a strategy like this, you don't have to develop as much in those defenses, right? Because you can just kind of go from cell to cell. You don't need to develop the defenses because the cell already thinks it's part of itself because it's hiding. It's wearing someone else's face, right? So that's kind of the strategy of an enveloped, in this case it's just the RNA virus, but a DNA virus would just have one extra step. It would have the exact same replicative cycle. The only thing here is that its DNA would first be converted into RNA. So you can only build proteins from RNA. So if it's a DNA virus, it gets transcribed into RNA first, which then gets translated into the proteins. But all those components of the virus doesn't make it. We make it. We make it out of our own nutrients. So because of this, we kind of describe viruses as being in this gray area of life, right? They don't have a way to reproduce without a host cell. And if the definition is DNA, half of the viruses out there don't even have DNA. They're just single strands of RNA, usually just a handful of genes in them. So all these things that do such kind of, that cause global pandemics are essentially just a handful of genes in one strand of RNA. But it makes all the stuff, those genes that it encodes, we turn it into, other COVID, other RNA viruses, or COVID viruses, if we want a kind of real world example of that. But yeah, the interesting point here is that really the only thing that kind of enters the cell is that DNA or RNA. Everything else, those proteins, the capsid, all the other parts of the virus don't even ever enter the cell. They're made out of our parts. And then they just use it to get to the cell, and then the only thing that goes in is that viral genome, that viral RNA. So knowing kind of what kind of goes in to a cell and what the cell itself makes, let's kind of go through this. It's kind of a trickier question, but on purpose. Let's see what kind of discussion this can stimulate. But okay, the slideshow thing isn't working for some reason. But don't let me exit. Okay, let me ask this iClicker question. Take a couple minutes to think of this. This is a trickier question by So if it's not obvious at first, I'll give you a couple minutes to think about it. But it basically describes bacteriophages, so a virus just like that spider-looking one. They were grown in a medium containing radioactive sulfur and phosphorus. The sulfur is incorporated into proteins, and the phosphorus incorporated into DNA. Now, if these phages were used to infect a bacterial culture grown without any of the radioactive isotopes, which isotope would we actually find in the infected bacteria itself? So before I kind of draw this out, I want to give y'all a minute to think of it just kind of in these terms. And then I'll draw it out if for those who are still kind of having trouble picturing this.
Is it
And then I'll give you another minute to think about it.
Is it phosphorus? Phosphorus.
Yeah. Bacteriophages grown in a medium containing radioactive sulfur and phosphorus,
I think it's phosphorus.
which are incorporated in the virus's proteins and DNA respectively, meaning the sulfur goes into the virus's proteins, the phosphorus goes into the virus's DNA. If these phages, these viruses, were used to infect a bacteria that was grown without any of those isotopes, without the radioactive sulfur or phosphorus, which one would we find in the bacteria itself? Sulfur or phosphorus? And so who feels like they need it drawn out? Y'all think y'all got it? I could draw it. Maybe don't want to show the hands, that's okay.
Is it
But, I mean, this is probably, this would be like one of the harder questions in a multiple choice exam. So basically what happens is, we're imagining a hypothetical lab where they're making a virus, right? So they're making a bacteriophage virus. Bacteriophage just meaning it's a phage virus, looking somewhat like this, some form of this. Sometimes it has the legs, sometimes it doesn't. And it's a bacteriophage DNA virus. It doesn't say it specifically, but the wording describes DNA. So I'll even just draw kind of like the helix. And so it says, contains radioactive sulfur and phosphorus, which are incorporated in the proteins and the DNA respectively. So that means the phosphorus, the radioactive phosphorus kind of goes in the DNA. So this DNA just has a bunch of radioactive phosphorus with it. Radioactive sulfur is in everything else, the non-genetic stuff. So that includes the capsid and all the proteins, all of these, the legs, the column, the capsid itself are made of proteins. And they're all lined with radioactive sulfur. So that's kind of the first part of this question. Then it says, if these phages were used to infect a bacterial culture, y'all should all know what a bacterial culture is at this point from 1102. Y'all grew your own with those lawn smears and the streaks for isolation. It's just a colony of bacteria kind of growing on a petri dish, for example. So just imagine a big petri dish. It's got agar, that stuff that helps the bacterial colonies grow. None of this has any radioactive anything. And so, one of these colonies kind of gets infected with the virus. And so, it's going to enter one of those bacterial cells. This is just the bacterial host of the bacteriophage, the bacterium. It's going to ask you, well, okay, now, which isotope would we find inside of this bacteria? Sulfur, radioactive phosphorus, or both? So now use that information, that sort of diagram, to see if that helps you answer this question a little better. So just take maybe 30 more seconds, think about it. Knowing what we know about the viruses and how they replicate in general. All right, most of y'all have responded. Give y'all just 10 more seconds. All right. Finalize your answers. And let's see. Okay, most support here for phosphorus, and that's correct, yeah. So the phosphorus are in the genome of the virus itself, and that's the only thing that will kind of go into the bacterial cell. All of the proteins get kind of disintegrated right away. Sometimes they enter the cell, but they do get kind of eaten up. And so the only thing that continues is that genetic material, which will get turned into proteins, create new viruses, but all those proteins are part of the host cell itself. So it doesn't have any isotope, any radioactive material in it. The bacterial cell itself will have some phosphorus because that DNA is indeed entering that cell. Okay, so let's go ahead and switch gears now to the next activity at this point. We'll talk about kind of we'll apply kind of what we've just lectured over to this next activity, and it focuses on just general viruses, their structures and replication. I say general because the vast majority of viruses share these characteristics we've been talking about. And then after this activity, we'll take a look at a sort of case study, looking at a specific type of virus that we've kind of heard plenty about in society. So let's go ahead and take 10 to 15 minutes on this one here, and then we'll kind of switch gears to a more specific type of virus. So with this activity, we're mostly kind of, some of it is a little bit more critical thinking, but some of it is just kind of exam questions. Give y'all a little more practice, give y'all an idea of how to talk about it amongst a group. See if as a team y'all can kind of tackle these multiple choice questions. How about team 1B? Would you call this an enveloped virus? True or false?
Oh, we said true.
True? Yeah. Yeah, this is basically the textbook image of an enveloped virus. Usually shown, you don't see the capsomeres, like those spherical subunits. It's indicating that this is kind of the host cell's membrane that the virus is kind of hiding within. How about team 2 close to me? How does an enveloped virus acquire its envelope?
You said A.
A, yeah, derived from that host cell membrane. It basically pinches out of the host cell, taking a piece of that membrane with it. How about team 2 far? Which of the 3 types of viruses, this one might have been tricky, shown in the figure below, would you expect to include glycoproteins as part of their structure? Did y'all come up with an answer for this one? For 4? D. Yeah. So did anyone else not put D? Yeah, so 1 and 2 here would require glycoproteins, kind of indicated by these pins coming out of here. The glycoproteins are one of the ways to enter a host cell. It kind of uses that lock and key analogy I sort of described, where basically a host cell has all these locks that allow you to enter it. And the host cell also produces lots of keys, but if you're a non-enveloped virus like the one shown right here, you still are producing potential keys that might bind to the locks of the host cell. And if they do bind to those locks, it'll allow you to enter the host cell. Number 3, though, kind of has that more EpiPen-like form of entry, where it just gets injected directly into the host cell. So these phages will basically burst a small part of that host cell open. No lock and key, no finesse about it, and the RNA or DNA will kind of be shot in from there. But these two rely on this lock and key, something we'll talk about more here in a couple minutes.
So number 1, is that glycoprotein or no?
So these protrusions, these things coming out, would be the glycoproteins.
Oh, because it don't say that
What's that?
in the picture in the book, it doesn't say glycoprotein.
Yeah, so when they talk about glycoproteins, I guess it's just, maybe it's implied. Honestly, I haven't looked at the textbook in a while, but.
Just say protein spike.
Protein spike, yeah, so the protein spike, we maybe have heard of that with COVID, right?
Mm-hmm.
That is a glycoprotein. That is essentially the way it kind of enters. And so some of them will be detected as kind of foreign. So the protein spike, when we detect it, if we have antibodies for it, we detect it as foreign and then we attack it. But some of them allow actually just entry into the cell too. But yeah, that term is glycoprotein. Was anyone able to order these steps? Any team want to take an attempt at seeing what kind of goes first? Yeah, Carlina.
First attachment, then replication, then assembly, and then release.
Yep, exactly. CBAD essentially. So first the virus will attach to the host cell, it enters, and then it'll get replicated. Basically what's being replicated are all the pieces needed to make a virus. From there, they'll kind of form together and assemble into a virus itself. So it's kind of like making a Lego structure, right? You have all the pieces being made, and then you put them together to build the final product. And then after that point, you release so that you can attach. And it's a cycle, right? So it goes CBAD right back to C. All right, kind of a longer one here. Was anyone able to tackle number 6 regarding the herpes virus? Which one kind of best describes these observations? How about, we haven't heard from Team 4. Were you all able to tackle question 6? Which one? D? Yeah, so the viral envelope mediates entry into the cell, the capsid docks onto the nuclear membrane, and then the genome is all that enters into that nucleus. So yeah, that's right. So the 3 main parts of an enveloped virus, it has those 2 things that every virus has, the genome and the capsid. It just has that 3rd thing, the envelope, which remember, that allows it to enter that cell. And then from there, the capsid docks to the nuclear membrane, and then the genome is the only thing that actually goes into the cell's nucleus and gets turned into the next generation, essentially. Okay, so another kind of along the lines of the concept check quiz we had with that radioactive isotope. A bacterium is infected with an experimentally constructed bacteriophage composed of the T2 protein and the T4 DNA. What would the new phages that are produced from this, so the next generation, what would they have? What did you all say?
What did it be?
B, T4 protein and T4 DNA. Did anyone else put B?
We did.
Yeah? Y'all put B? Yeah, that's right. So even though it has protein from something else, remember that the DNA, the genetic material, is the only thing that actually goes in. So you could have, you could call the protein in this first bacteriophage, you could call it anything. It could be the lollipop protein, and it's not going to be converted. Whatever is in that DNA is what gets converted. So the T4 DNA is going to make T4 proteins as a result. So the next generation, even though that first generation had T2, the next generation will be all T4. It's whatever the DNA says. I think we've driven home this point of comparing and contrasting lytic versus lysogenic. Lytic is a bit more brutal. The cell essentially dies immediately as a result. Lysogenic is a bit more insidious. The virus's genome gets incorporated into that host cell's genome. We'll learn about prophages here in a minute in the last part of this. How about 9? Emerging viruses. So COVID was one of these emerging viruses. We've heard of other ones as well. All these pandemics that are virus-based were at some point emerging viruses. What did you say? A, mutation of existing viruses. D.
All of the
Okay.
above. Right?
Yeah, so you're a third right. A is right.
All of the
So is
above.
B and C.
Yeah.
The only one that isn't right is E, right? So all of the above. So a virus can mutate, and then it switches essentially maybe from that mutation. It goes to a new host species. So A and B often are kind of intertwined. Or the virus could have existed in a really isolated population, and so nobody ever really knew about it, and then some, the population gets urbanized in some way. Starbucks shows up, people start coming in droves, and then the virus spreads from there, right? A population that's isolated with the virus meets the rest of the world, and then it spreads rapidly from there. So yeah, D, all of the above. All right.
Airplane travel.
Airplane travel, exactly. If you haven't seen the movie Contagion, then that, you should watch it. It actually is a pretty good explanation of kind of viral transmission.
Train to Busan.
What's that?
Train to Busan.
Oh, Train to Busan. Yeah, I haven't seen the second one, which came out last year, but the first one is awesome. Yeah.
There's a second one?
There's a second one, yeah. It looks awful. But the first one is awesome. Yeah.
The first one was correct.
Yeah, very good movie. Yeah. I want to switch gears here now. So we've talked about kind of general viruses. Now what we'll do kind of for the last few minutes is we may not have time for the activity, but we'll kind of start right after the IRATs and TRATs and talking about the exam on Wednesday. We'll move into this activity, but I want to kind of give you a couple of precursors into this. So we'll talk about HIV, but I also want to play a video before this. Not necessarily about HIV, but a different sort of case study. Oh, no, not this one. There we go. So obviously we've heard a lot about COVID over the past few years, and we're pretty up to date on its transmission, how new variants are always popping up, and new vaccinations to address each of those variants. One thing we often see in the media, especially towards the beginning, was how it compared to previous pandemics and epidemics. People were always trying to see, okay, which one was deadlier. One thing we often, one of the ones that we hear about the most is perhaps the influenza pandemic of 1918, which was also referred to as the Spanish flu. You might have heard of that. So I want to show you this video just to get a sense of the kinds of numbers we were dealing with back, in the days of this flu about 104 years ago. And also give you a sense of how it was transmitted, and then we can relate it back to some concepts you read about in the book and saw in class today. So let's go ahead and watch this first.
One of the worst natural disasters in recorded history was caused by a virus. The influenza pandemic of 1918 struck every major U.S. city, dropping people where they stood. There were at least 20 million people killed worldwide, and it's probably the worst pandemic the world has ever known. There were more people killed from influenza than there were from the First World War. The war itself was the problem. If there had been no war, that year's strain would have stayed isolated. But when Allied troops met in northern France to exchange fire with the enemy, they also exchanged a lethal strain of flu. So it began. It then went from France to the United States and disseminated from Boston. At Fort Devens, Massachusetts, one case of flu turned into more than 6,000 cases between September 1st and September 18th. It took only 17 days. India lost nearly 4% of its population. Alaska, 8%. In the South Seas, the death rate was 20%. People were absolutely panicked because somebody could be playing bridge one night and they would be dead the next day. And so they did all kinds of things. They were wearing face masks and, in fact, the police all wore face masks. They were carrying little pouches of garlic and onion. Just anything that they thought would protect them from the flu. They had no idea that they were leaving exposed the very place where flu enters the body, their noses.
Oh.
Nothing was known about how to cope with it. And that is something that began to emerge when we got into the 1930s, 1940s, 1950s. By that time, there was enough knowledge and experience and the development of techniques to make it possible to proceed.
So just a short clip there, but there were some similarities we might have seen from that to kind of today, right? In general, this was an isolated incident until it wasn't, right? So if y'all can kind of remember from now, 2.5 years ago, I don't know if y'all remember kind of January and February of 2020, we were talking about, looks like China's shutting down and nothing had made it here. And then it was, okay, there's a couple instances and I think it was San Francisco or somewhere in the Pacific Northwest. And then suddenly, the NBA shuts down. And that was the moment for me that I was like, oh, okay, now this is serious. Like, the Rockets aren't playing anymore. And yeah, then kind of the world changed from there. But as we watch this video, we kind of see some similarities, right? There were some strategies that were applied then that are applied now, right? Masks, isolation, two strategies that kind of stuck. Others, not so much, right? They were talking about wearing garlic and onions. That nobody was saying to do that now because we have a little bit more knowledge of how these things are spread now. So keep in mind, this was before vaccines, really, before a sort of, this was a largely pre-vaccine era that this pandemic was occurring in. And it was particularly harmful because, it was specifically targeting actually 20 to 40-year-olds. But, just like with COVID, it was isolated kind of to this northern region of France until it wasn't. And it's believed that it was actually kind of had been in that northern region of France for years. With the case of COVID, we were talking about, China isolating because of, the Wuhan region where there were a few cases. And so, that region, from that region to the whole world was only about a month or so. The Spanish flu, which, screw that name, right? This happened in France, and Spain wasn't even there during World War I, and still, I guess we got blamed for it. But the Spanish flu, it had existed in that northern region of France for at least 2 years, it's believed. So you can see really what this kind of increased travel throughout the world amongst people has kind of done. It kind of exponentially raises the transmission rate of these diseases that could just be isolated incidents. But that it's very, very hard for anything to be isolated. So, just wanted to show that as an example. When we're talking about these pandemics, largely it tends to be a viral pandemic. Part of that has to do with how quickly viruses can evolve. So we've seen the multiple strains of COVID, right? Each one of those strains is essentially a natural selection experiment at work, right? A vaccine is created and applied as best that it can be, and it targets a certain strain. Well, all the individuals that are remaining are largely resistant to that strain, so they become the next strain. And so you build a vaccine to respond to that. Our vaccine efforts are often responsive, right? They're reactive and not proactive, right? So it's hard to predict which strain will become the widespread one. So we respond in light of that. So for the last couple minutes, I'm just going to give you a brief description of one virus, HIV, the human immunodeficiency virus, which we often associate with AIDS. And we'll get a kind of better understanding today and on Wednesday about what the relationship is between HIV and AIDS itself. AIDS is actually just kind of a symptom of HIV. It seems kind of almost rude to call it a symptom, right? Because it's a very grave situation. But it's just to mention that it's a syndrome that you get from HIV. And basically, the way it works is similar to a replicative cycle that we've already seen. So HIV specifically undergoes what's called a lysogenic cycle. So not the lytic cycle, but the lysogenic cycle. So let me zoom in here because I know this is getting really hard to see here. But we can sort of follow an HIV virus as it enters. It has its glycoproteins around an envelope, so it's an enveloped virus, and it'll use that envelope to enter a host cell. Its RNA will get transcribed into DNA, which will then enter the nucleus and become part of the host cell DNA. So individuals with HIV, one of the reasons it's so difficult to kind of rid yourself of the virus is it's undergoing this lysogenic cycle where it goes into your own genome. Yeah.
Does it bind to leukocytes?
What's that?
Does it
it's going to attack T cells is what it attacks. But yeah, what it largely does is goes through this insidious replicative cycle where it's within your DNA. Some of that DNA we call this a provirus when it's kind of embedded in your own DNA, the provirus. And then RNA will be made from that, which makes other HIV molecules, and it undergoes that same process that we've already seen before. But the only difference here is this sort of lysogenic process where instead of RNA being turned right into the proteins, it gets turned into DNA and becomes part of your genome. It gets incorporated into the host cell DNA as well. And then, yeah, just like you said, HIV specifically targets T helper cells, so these CD4 cells. And you can actually see it under a microscope when you look at the individual cells. When it's healthy, it kind of looks like this blue orb. An infected T cell is full of basically all of these HIV particles that are sort of plaguing it. You can almost see that it looks relatively sick. And the stage you get from HIV to AIDS is basically just HIV has weakened your T helper cells to a point that they just can't function. Your immune system isn't functioning very well to fight the infection. So you can see how these T cells are impaired in some way. So AIDS is just kind of, it's considered AIDS after a certain point. But HIV and AIDS kind of occurs along a continuum where over time the immune system is kind of getting more and more battered. But what I want to talk about specifically for the last couple minutes is how they enter immune cells. So I talked about this key and lock mechanism, I'm just going to go to the board one more time because with HIV, it's a sort of more complicated lock that HIV is taking advantage of. And what we'll learn about on Wednesday is how mutations have actually helped some individuals work around this HIV infection. So if we imagine the host cell, we'll just draw the surface here, kind of the surface of the capsid HIV, and it has one of these keys. And what it's a key to is a lock that's made of two proteins for the host. And those two are the CD4 and CCR5. So basically what happens is you've got stuff here, but we'll call it CD4, and then this second block here we'll call the CCR5. So they usually occur and where the CCR5 will kind of form right next to a CD4, and it forms this gap, and it's meant to bind to something and let it in. The HIV essentially just takes advantage of this lock, but it's a lock that's made up of two components. So it has a nice fit, and when it binds to this CD4 and CCR5, it triggers the cell to kind of open up and accept the HIV molecule, the HIV virus, after which it'll kind of undergo its cycle. So there's an intricate lock mechanism here that's going on. And what's interesting, and what we'll read about, are individuals who have a mutation that actually just makes this CCR5. It's called delta CCR5. And it basically makes a non-functional CCR5. Specifically, it won't make CCR5. So individuals with this mutation actually don't produce CCR5. And so what happens is individuals with this mutation, I don't want to say are immune entirely to HIV, but the main mechanism by which HIV enters a host cell is blocked off. This doesn't function as a lock anymore, and so HIV molecules actually don't enter host cells through this mechanism when the host cell is one of these mutants. They don't produce the lock, and so some individuals, they've been kind of studying the genetics of it to try and make kind of cures and vaccines for HIV, and they rely on mutations. This is just one of the guys, but using bone marrow donations, trying to get see it as a mechanism for curing HIV specifically, because if you kind of knock out this main mode by which HIV enters the cell, you can kind of hopefully forego at least the most deleterious stages of HIV/AIDS, right? So we can use genetics to better understand how this whole process works. But essentially, mutants without this, the protein product of this gene, don't seem to have HIV enter the cells, at least not as in as abundant forms as those, the majority of the population that does. And so what we'll talk about on Wednesday, after the IRATs and the exam stuff, is why it is that it's just a small population that have this mutation within Europe. So we'll see what it has to do, what's so special about these Europeans that they have this mutation. So yeah, that's all I got. I'll stick around if you have any questions, but yeah, otherwise see you Wednesday. We will have the IRAT Wednesday.